Welcome to cellular structure. This is getting to know the cell. You want to learn the structure and the function of each of the organelles. First, I want to take a look at the cell theory. All living things are made of cells. Cells are the basic units of structure and function of an organism. Cells arise only from pre-existing cells. It's really important to realize how strongly the function of the cell determines the shape. You've heard a lot about stem cells in the news and things, and these are cells that are actually able to differentiate into all other cells. Here is a picture. This would be the fertilized egg or zygote, and as it starts dividing, when it gets to just beyond the blastocyte, and it's still completely undifferentiated from all of those specific cells, they can be differentiated into any of the other cells. Pretty important to be able to do a lot of research going on right now with those. White blood cells. White blood cells are able to change the shape to get where they are needed. So if we take a look at the white blood cell, the white blood cell, right now you really can't tell that, but let me show you this quick video. Those that have had my class before should be able to remember this. Okay, this is a white blood cell. This is a white blood cell chasing some sort of larger bacteria, and notice how it's moving around. Move and push those other red blood cells, and its shape is able to change. Notice how it just kind of engulfs it. That's called phagocytosis. Next, we have the nerve cells. These have long extensions to quickly relay messages. Here's the center of the cell. You see these long extensions. So they're able to connect to other nerve cells and able to quickly run from one nerve cell to another, all these electrical impulses. Structure very important here. Skin cells are flat to be able to protect. This has been dyed. A fluorescent dye, but these are all skin cells. These green ones here, the green actually represents keratin, the red is the nucleus up in here, and here they are as they are differentiating to start filling up with the protein keratin. Notice how thick those layers are. And I'm sure you remember that cells are the smallest units of life in all living things. Here is a diagram of different pictures of different types of cells. You can see all the different structures. These are just drawn. But notice how different all the structures are in the shapes, and those are very specific for the functions they have. The nerve cell. Here are the red blood cells, and here's the white blood cell that we looked at. Reproductive cells, bone cells, just all different shapes. Those different cells come together to form tissues. Here are a bunch of bone cells to form the bone tissue. These are cartilage cells. We will look more at these next week. Different blood cells. Here's again is some more nerve cells, which makes up nervous tissue. And these are long individual muscle cells. And they are multinucleated. So all these cells look differently. Now we're going to take a look at a picture that's just a basic cell that has kind of a compilation of all the other organelles. So reminder back to basic biology, which one of these is a eukaryote and which one is prokaryote? Eukaryote is much more complex. You are a eukaryote. Notice there is a membrane around the nucleus, and you have all these different organelles that help carry out specific functions, and we're going to take a look at these different organelles. First one is the cytoplasm. The cytoplasm is everything between the cell membrane, right here, along here, and the nucleus. So that includes all the other organelles. So the cytoplasm is all the liquidy stuff called cytosol that's inside and surrounding, and all the organelles between the nucleus and the cell membrane. And there is the plasma, or the cell membrane, sometimes called the phospholipid bilayer. Very specific job. It encloses the contents of a cell. It selectively allows certain things in and certain things out. It regulates those substances through the membrane, actually those phospholipids. And remember the picture in the diagram of those proteins that were also in there, embedded in there. So mainly you just need to remember that this is very selective. The cell membrane is very selective in what it allows to go into and out of the cell. This helps to maintain homeostasis. Phospholipid bilayer. Next, you have the cytosol. The cytosol is actually just the liquid, that gel-like fluid inside. Cytosol would be bonus for y'all because I don't have that specifically listed. But this is the cytosol. This is, majority of it is water, but it is filled with all different molecules, and these molecules are continually going through chemical reactions. Remember, we talked about how water is so great for being able to separate out ionic compounds, so those chemical reactions are going on here, and all different proteins are being shuffled around. These are the ribosomes. Notice where they were located at. All right, we have free ribosomes, and these ribosomes specifically were on the endoplasmic reticulum. And the ribosomes actually make or synthesize the proteins. Remember those strands of amino acids that get folded up? So they are considered the protein factories. And we're going to look at this one again. We're going to say these little dots are this gray here, and this is a ribosome that is putting together, linking together amino acids to grow proteins. Sorry about that. Next, we have what those ribosomes were on. This is the rough endoplasmic reticulum. It is considered rough because the ribosomes are on there. Now, this is an extensive system, and if you will look back, notice how it is connected to the nucleus. That is because it is going to help proteins. It is going to make these proteins on the ribosomes, and then it is going to move those proteins throughout this system and help with the folding to be able to form the proteins. Next, we have the smooth endoplasmic reticulum. Now, same endoplasmic reticulum, but yet it does not have the ribosomes, so it will not be making the proteins. Instead, this is going to synthesize or make the lipids and fats. Remember the lipids? Those long hydrocarbon chains that had saturated and unsaturated. We had the phospholipids in the bilayer. We had different hormones, testosterone, and estrogen. So these would be made in here in the smooth endoplasmic reticulum. Now notice the difference between the Golgi body. It looks very similar to the smooth endoplasmic reticulum, but yet it has these buds normally. So look back at the smooth endoplasmic reticulum and you'll see this budding off. It's like little pieces are going to come off of the Golgi apparatus, or sometimes it's called the Golgi body. Okay, so we still have this extensive system of all these canals that proteins are going to be moved through. So you'll have proteins that will be shuttled over here. They will continue to be processed. In other words, they will continue to be folded up in the right shape. Then they call it packaging and distribution center. So you will have these proteins that will end up here on the ends of these, and they will bud off and actually make a little vesicle around them. Then it can be shuttled either outside, the proteins can be shuttled outside the cell, or be used and taken over to other parts of the cell. So the Golgi body, or Golgi apparatus, is going to take proteins, and it will take lipids also, and some of the fats, and continue processing those molecules, and then package them off. These are little packages, and then distribute to them either outside the cell or into over to another area. These are the centrioles. The centrioles are number 14 on yours. Now, your list says, and your book says, it's the formation of basal bodies. But instead of that, I want you to remember, more importantly, that they help the cells divide. If you think back into mitosis, here's a quick picture of mitosis. You can see that we have these centrioles, and they are moving around to the opposite ends. And remember, they are going to send out and reach out and attach to the middle of the chromosomes at the centromeres and pull those chromosomes apart. So that is the job of the centrioles. So you can see how important the centrioles are in helping cells divide. This isn't really a very good picture of the cytoskeleton. Sometimes we refer to it as the microtubules. You can kind of think of it as, and you probably don't remember Tinker Toys. They're big, long spaghetti noodles before they've been cooked. These help with cell shape and actually the movement of different proteins. And so if you have these hard spaghetti noodles laid throughout the cell, then you can, so it's here they are, and so if they were all throughout the cell, you can see that it could help with the structure, holding the shape of the cell together, and then also allowing, remember I said the proteins that bud off of the Golgi body in those little vesicles, those can then travel along the cytoskeleton, and so it helps the movement of different organelles. Next, we have the lysosomes. Now, the lysosomes are referred to as the killer organelles sometimes. They have digestive enzymes or proteins that are in here that are going to act as enzymes to help break apart old organelles or different things that come in, say bacteria or viruses that come in that need to be broken down and disposed of. So it's considered intracellular digestion. So they stay inside the cell and they help break down different organelles and other items that come in to the cell. Next, we have what would be considered cilia, and these are kind of hair-like structure, and flagellum or flagella. Now, both of these we say enable movement, kind of to be able to differentiate them. Cilia is much shorter, and cilia will line this is on the outside of the cell, and cilia will line down through our esophagus and help food pass down. All right, it also can go up through our nose area and down through the back of our throat. and will help capture foreign bodies and then help them either move on down or move on back out. So they still enable movement, not necessarily the movement of the cell, but the movement around the cell or things on the outside. This is actually a picture of cilia that is lining our lungs. So you can see it looks like little short tufts of hair. These are able to capture impurities and move them up and out of the lungs. And I did a terrible job on the flagellum because in the human body we will never have more than one flagellum, and there's only one type of cell that has flagellum, so I've got to change that. So just imagine all of these whited out and one long one. Here, let me show you a great picture of the only cell, the only type of cell that has a flagellum. Flagellum does help this cell, particularly this cell, to move. All right, and guess what it is? Yep, it is a sperm cell. So in the human body, the female does not have any cells with flagellum. Only the males do. All right, it is just about time to move on to the second part. Thankfully, the second part is only 6 minutes long, so it's a lot shorter than this one. And you will start off with the nucleus. It has a little bit of a dead space, a dead quiet time in the very beginning, but just give it about 30 seconds and it will start.